Good evening, everyone. Hope you can hear me. My name is Kambiz Ranavardi, co-president of ColumbiaDC and a graduate of School of Engineering and Applied Science. We are very honored to have Dr. Dietrich Egli, assistant professor of developmental cell biology, and Dr. Sam Sternberg, assistant professor of biochemistry and molecular biophysics at Columbia University, to talk about CRISPR, its applications, and related pioneering research at Columbia in preventing heritable diseases. Dr. Francis Onyimba will be our host this evening. Please allow me to very briefly introduce our storied and incredible panel. Dr. Dietrich Egli is Maimonides Assistant Professor of Developmental Cell Biology at Columbia University Irving Medical Center. He is one of the founding members of the New York Stem Cell Foundation Research Institute, where his group made numerous advances in somatic cell nuclear transfer and mitochondrial replacement. His area of research includes human embryonic development, somatic cell reprogramming, cell cycle regulation, and double-strand break repair, and preventing the transmission of disease-causing mutations in human reproduction. Dr. Sam Sternberg runs a research lab at Columbia University, where he is an assistant professor in the Department of Biochemistry and Molecular Biophysics. His PhD in chemistry from UC Berkeley was under no other than the mentorship of Nobel laureate Dr. Jennifer Doudna. Sam's research focuses on the mechanism of DNA targeting by RNA-guided bacterial immune systems, and on the development of these systems for genome engineering applications. He's the recent recipient of the NIH Director's New Innovator Award, and is a Sloan Fellow and a Pew Biomedical Scholar. And our host, Dr. Francis Onyimba, is gastroenterologist at the University of Maryland Medical Center. She completed medical school at Columbia University College of Physicians and Surgeons prior to completing her internal medicine residency and a fellowship in GI mobility and neurogastroenterology at the Johns Hopkins Hospital. In 2019, she was selected into the Young Physician Leadership Scholars Program by the American College of Gastroenterology for leadership development and physician advocacy. Without further ado, Francis, take it away.
Great. Thank you for that introduction, Kambiz. Hello to everybody who's watching tonight. It is my pleasure to join you all tonight. It is my pleasure to be amongst scientific greats like Dr. Sternberg and Dr. Egli. I am quite excited for tonight's discussion about CRISPR. Those of you who joined us earlier this year for our virtual events may remember we had an event about the ethics of eugenics and gene editing in embryos. Tonight, we're going to take it a little bit broader and at the same time do a deeper dive into the science behind CRISPR and CRISPR-Cas9 technologies. What is CRISPR? How does it work? What are the therapeutic applications? And then what is the role in reproduction? So it's not about me. I'm here to really just keep us on time. So without further ado, I'll pass it along to you, Sam Sternberg.
Wonderful. Thanks, Francis. It's wonderful to be with all of you today. So I'm at Columbia now, but I am also a Columbia alum of Columbia College 07. So I hope we can have a great discussion later today. Let me share my screen here. Okay, so hopefully not too much. This is going to be a refresh from what you've already heard about CRISPR earlier in the year. But I thought I would really take a step backwards and really get to where does this CRISPR technology come from, and how was this CRISPR conundrum initially cracked to open up this gene editing revolution. And I think fundamentally, in the lab, we are thinking about how the DNA of an organism leads to all the emergent phenotypes of cells, of multicellular organisms. And this is going to be where gene editing has its greatest power, but it's also really kind of fundamentally where the CRISPR discovery started, thinking about what kinds of sequences in the genome of simple bacterial organisms protect themselves from pathogens. Now, as Kambi's mentioned, of course, CRISPR has been in the news and certainly all around the scientific world since at least 2015. You know, I started working in Jennifer's lab before CRISPR had gone mainstream in the science world. And now, you know, I think this past year was one of the most exciting for me, seeing the Nobel Prize finally awarded to Jennifer and Emmanuel. Some of the first clinical trials showing incredibly promising data for sickle cell cures in the first US patients. You know, I think we're really at this inflection point where the future of medicine for certain genetic diseases and cancer may really be changed with the advent of these gene editing approaches. There's been an amazing explosion of excitement around this technology in the biotech world. Many companies have started developing CRISPR technology. Many companies, you'll notice, have been around for decades and have now brought CRISPR platforms into their own company. But you can, of course, also go to the public media. I don't know how many of you saw Rampage. It was actually one of my first lab social outings back in 2018 when I started in New York. The Rock, Dwayne Johnson, talking about CRISPR on the big screen in the Harlem Magic Johnson theaters. It's been on the X-Files, and you'll see CRISPR splashed across the front pages of the New York Post, Time Magazine, The Economist, because of this kind of awesome power to change evolution at its core, really. The ability to make targeted, rationally designed changes in the genome. And what does that really mean for disease treatments, but also maybe for reproduction? So I think it's going to be really fun for me to dive backwards on CRISPR and then have Dieter bring us up to the present. And of course, he'll, I'm sure, cover the kind of game-changing news in 2018 that the first babies were born in China who had their genomes modified with CRISPR. But I'm going to take us back about 30 years in time into unpacking this kind of complex topic. Where did CRISPR come from? Why was it even discovered in these bacterial genomes? And then what was that major inflection point to take this CRISPR biology research into the realm of gene editing technology? And so I'd love you to come away from this first 20-minute talk appreciating that there's really this divide between the way that scientists like Dieter, like myself, are applying discoveries made about CRISPR to modify genomes, but this very, very rich and critical research into the natural way that CRISPRs function in bacteria. So CRISPR was not invented in the laboratory. It was something that was sourced from bacterial genomes, from the way that bacteria fight off infections. And by understanding how that works on a molecular level, that really gave us this new toolkit to modify DNA in a very synthetic engineered manner. So that CRISPR discovery story starts back in 1987. This is kind of the first paper about CRISPRs before the acronym existed. This was a Japanese group. They were studying some genes in E. coli. And although the focus of their article wasn't on these CRISPRs, in the last paragraph of their paper, they commented on having come across what they referred to as an unusual structure of DNA. So they had one figure kind of pointing out this oddity. The last sentence, they mentioned that the biological significance of these sequences is not known. And really for 20 years, that remained a mystery. What are these unusual structures in the DNA of E. coli doing? So in my laboratory, we spend a lot of our time looking at bacterial genomes, doing comparative genomics to try to piece together an understanding of what different regions of the genome are kind of imposing for how cells protect themselves. And here we're actually looking at a zoom in of that E. coli genome. And this is the unusual structure that those Japanese scientists first commented on. These highly repetitive sequences where every yellow highlighted region is the exact same 30 letters of code over and over and over again. This is something that really hadn't ever been seen before. But by the mid 2000s, what was even more surprising is that about half of all bacteria on Earth whose genomes had been sequenced had the exact same structure. So clearly, evolution had maintained this type of repeating element. What was its function? Well, before we get to the function, of course, we have this colorful acronym that was coined to describe this characteristic pattern of clustered, regularly interspaced short palindromic repeats. That's the CRISPR word. So in terms of what these are doing, the first clues there didn't come until 2005, when 3 different groups appreciated that it's actually not the repetitive sequences that are interesting, but the little snippets of DNA tucked in between them. So these groups found that if you now analyze the DNA sequences interspaced between the repeats, in many cases, these were a perfect match to viruses. Now, we've heard a lot about viruses in the last year. Some of you may be surprised to learn that bacteria are infected by their own types of viruses called bacteriophages. These are actually the most abundant biological entity on Earth. They outnumber even bacteria about 10 to 1. So everywhere we think about finding bacteria on our skin, in our guts, in food, in the soil, they are constantly being attacked by bacterial viruses. These are actual electron micrograph images of a cell, in this case shown in the bottom left, being bombarded with these viral particles. If we look at a more cartoon view, these in some ways are kind of analogous to the coronavirus in the sense that they have a protein exterior that packages the genetic material inside. In the case of the virus shown here, it's DNA, although some viruses can have RNA as their genetic material. And these viruses latch onto the cell. They have pumps at the base of the tail that literally forcibly inject the DNA inside. And in the event of a successful infection, the cell is burst open while spewing out dozens or hundreds of additional viral particles that then go on to infect the surrounding cells in that environment. So by the 2000s, of course, every molecular biologist knew that bacteria have these viral pathogens. And we thought we kind of knew most of the immune systems that bacteria employ to protect themselves. But then here comes this major question: are these weird repeat sequences and the snippets of viral DNA in between them a new kind of immune system? And so that breakthrough discovery that proved exactly that hypothesis came from, of all places, a yogurt company. This is a company studying how the bacterium Streptococcus thermophilus immunizes itself against viruses. Why would you care? Well, this is a $40 or $50 billion a year industry. You don't want to have your bioreactors collapse because your cultures are getting infected. And so they were studying how these strains maintain resistance. And they found that anytime a strain had a matching snippet of DNA that had a match in the CRISPR, the bacterium was completely immune, proving that these CRISPRs are conferring adaptive antiviral immunity. So effectively, the bacteria have something akin to a vaccination card that they store in their own genetic material. I'm sure many of you have gotten your COVID vaccination card in the last couple months, I hope. Bacteria are doing the exact same thing, except they are storing the memory of that vaccination in their own genome. And then they can use this sequence of DNA in future infections to recognize, well, hey, this DNA is not my own, it's from a virus. Now I need to go and neutralize that threat. So if we think about that kind of metaphorically, it's really not so different from what the Department of Homeland Security might be doing by storing fingerprint information, retinal scan information. It's giving individuals the ability to recognize a foreign threat and prevent them from maintaining some kind of offensive action in the country. Bacteria do this using DNA as their identifier, and they essentially prevent the phages, the viruses, from infecting them. So the focus of much of my PhD was really understanding how does this work on a molecular level? What are the molecules involved? And that really led to the discovery of CRISPR-Cas9. So you can think of it as being like a pair of precision-guided molecular scissors. The bacteria use these scissors to target viral DNA. The DNA gets initially cleaved or cut into 2 pieces, and that triggers the eventual destruction of the rest of the viral genome. Here's a more realistic view of how this CRISPR-Cas9 looks. In gray is the protein component, and in black is a molecule of RNA. And the RNA is really what provides the GPS coordinates telling the enzyme where to go. And the Cas9 enzyme is the weapon. It's the arsenal that is deployed to go and find the matching DNA sequence and cleave it. So if we think about what this looks like kind of in real time during an infection, here's that bacterial virus latching onto the outer surface of the cell. At this point, the DNA gets injected, and the cell has about 15 minutes to find that DNA and destroy it before the virus replicates itself and destroys the cell. And so here's our CRISPR-Cas9 molecular machine coming into action. Again, the protein component is a big blob. We have the RNA component here in red, and this complex goes around the cell looking for matching sequences to the guide RNA. Those sequences are identified through base pairing, and then the weapon swings into action and severs both strands of the DNA. So this discovery is really what won Jennifer and Emmanuel the Nobel Prize, because it immediately kind of opened up this opportunity to take these same biomolecules and repurpose them for something quite powerful. And that brings us back to this transition point, this disruptive idea of repurposing these CRISPR molecules for gene editing, where rather than bacteria using CRISPR to cut and destroy viral DNA, we, the scientists, can repurpose CRISPR to cut human DNA, but use this to actually modify permanently the underlying sequence. So Dieter will kind of cringe probably seeing this slide here because of how oversimplified it is. So he's going to tell you more about what's really going on. But at a high level, the reason that we can use CRISPR-Cas9 for gene editing is because after making this cut in a human cell, this DNA break is recognized by endogenous enzymes that actually trigger the cell to repair that broken DNA. And in some cases, although not all cases, the scientist, we can actually coax the cell, we can trick the cell into repairing that broken site using something called a repair template, where we now induce the cell to glue together those broken ends, but replacing the original sequence with a new sequence of our own choosing. And so what CRISPR is really allowing here is the ability to make a very specific lesion at a defined site in a chromosome of interest. And then the cell is going to recognize that break and glue the ends back together in a way that leads to a permanent edit at that one region of the genome. So again, Dieter's going to fill in a lot more details there, but this was the idea. We're going to use the cutting part of CRISPR and the repairing part of the human cell or bacteria, an animal cell, and that's going to give us this ability to do gene editing. Again, from a metaphor, we can think about this being very similar to find and replace. We have a search term, that's the guide RNA. We have a replace term, that's that repair template. And CRISPR is going to basically allow us to get from one to the other. And we just need to do this not in the language of the alphabet, but in the language of DNA, those 4 letters A, G, C, and T. So 2012, we have this kind of breakthrough idea of using CRISPR in exactly this way. In the next 6 months, we have a massive race to do it first in human cells. Of course, I'm not going to talk about it anymore, but there's a huge patent award that spun out of exactly those kind of 6 months of fervor to get it working first, and that's still ongoing. There's billions of dollars at play here on the many licenses coming out of this technology. But a number of groups showed in early 2013 that indeed these CRISPR molecules do work in human cells for gene editing. And then in the years that followed, there was kind of an explosion of applications repurposing the same exact molecules in just about any model plant or animal species that you can think of. Major crops like corn and rice, major animal models like mice and rats. And then of course, in 2016, the first clinical trials began for cancer treatments using gene edited immune cells. So I always like to point out CRISPR is not the first gene editing technology, but the reason it has exploded in this kind of exponential growth is because it's very easy to use, it's extremely inexpensive, and there are nonprofit companies that globally distribute the materials that are needed by any lab in the world to perform their own gene editing experiments. So it's kind of the classic disruptive technology in that it really lowered the barrier to entry to a point where any laboratory that does standard genetics can now use CRISPR. And if you go to a place like Columbia or Johns Hopkins or University of Maryland, you're going to find CRISPR probably being used in just about any lab that's doing some kind of human genetics research. And so in the last couple minutes, I'll just highlight a couple major areas where I think CRISPR is being applied that are going to really impact society in our lifetimes. And then I think we can imagine a little bit what might it look like in the next few lifetimes coming down the line. So I think what gets sometimes the least attention, but it may be the most important application area for CRISPR is just basic research, because now we have the ability to make extremely precise changes to DNA and ask in a very well-controlled setting, what effect does that have on the emergent traits of the organism? We can ask kind of esoteric questions like what genes are involved in a butterfly having certain wing coloration patterns, what genes are involved in axolotls or Mexican salamanders regenerating their limbs, but in a more kind of direct disease relevant manner, what genes allow a cancer cell to become metastatic? Using a very powerful genome scale type of CRISPR experiment, researchers are now kind of interrogating the role of every gene in the human genome to understand what allows a cancer to become more proliferative when it has certain types of mutations. And in this case, CRISPR is not really going to directly address the cancer therapeutically, but it's going to tell us what kinds of new drug targets should we be going after. And then kind of very relevant to the past year of the COVID pandemic, there's been a huge amount of research using CRISPR to understand what genes is SARS-CoV-2, is this coronavirus, absolutely dependent on to maintain an infection. And if we understand those genetic susceptibilities of the virus, can we now think about new ways to kind of treat patients with this infection based on this genetic knowledge? In the context of agriculture, I think this is an area where you're going to probably find CRISPR kind of out there in society first in grocery stores, where I think the ability to do scarless gene editing is forcing us to rethink what does it mean to be genetically modified. These crops shown here and this cattle, these are not GMO in the classic sense. There's no foreign DNA in the genome left behind. We're using CRISPR to make single letter changes. The CRISPR gets lost, but you're left with some potentially enhanced major food staple. And I think there's a lot of interesting research and regulatory decisions being made in the last couple of years on how will these new kinds of products be administered and be controlled. And then, I think really where there's incredible excitement is using CRISPR for medicine, where we now can take this knowledge that has been developed over the last couple of decades for diseases like sickle cell, cystic fibrosis, Huntington's, where we now know the exact region of the genome that is mutated, that causes these diseases. Can we now take gene editing and actually repair these diseases at the level of DNA? Or in cancer, can we use gene editing to enhance the ability of immune cells to go find and fight cancerous cells in the body? These are kind of the two major areas where clinical trials have already begun, where I think we're going to really see some changes to medicine over the next decade. So what's possible today? In a laboratory environment, it's become quite straightforward to correct these mutations. So you can find hundreds of articles now that have shown in cultured human cells growing in a petri dish, very straightforward to use gene editing to completely repair or reverse the causative mutation. The challenge is, how do we do this safely and effectively in the complex human organism, where we have to wrestle with the whole range of other safety concerns, things like how to deliver it into the body without provoking an immune response, how to edit the right tissues, the right organs, and how to avoid all of the possible adverse side effects of this type of treatment. I think there's incredible promise. This was the first story of gene editing actually saving a girl's life. This was a patient in the UK who was dying from leukemia. She received an infusion of gene-edited immune cells and was essentially cured of her cancer in the process. And so just as a brief update, although you can read plenty more online, the first trials for sickle cell and beta thalassemia have begun. There's some remarkable data coming in from these patients that have now been transfusion free for multiple years. So I think, you know, the idea of actually curing these diseases is very much in sight. And then we have a number of clinical trials for cancer immunotherapy, as well as the first trials to actually treat diseases in vivo, where we deliver CRISPR directly into the body. Now, I do want to stress, although there's incredible promise, there are a number of challenges that are going to be obstacles before that promise is fully realized. Dieter's going to talk about some of them, but again, delivery, really understanding what kinds of edits are being made, how can we avoid off targets, kind of introducing inadvertent mutations elsewhere, and then things like the body rejecting CRISPR because it's foreign. I'm going to leave this topic all for Dieter, but I think, you know, you can think about where in the body do we apply CRISPR, what kinds of cells would we want to modify, and we can do that on body cells like the blood stem cells or the eye. We can also do that on germ cells. But what I wanted to end with is kind of where my lab spends a lot of its time thinking, which is there's more tools out there. In the same way that there was these kind of foundational studies of how bacteria fight off viruses that led to CRISPR-Cas9, there's a whole lot of other CRISPR systems out there waiting to be discovered. And so if you think about Darwin's classic tree of life drawing, there's a tree of life for CRISPR where all of these different spokes represent new evolved versions of CRISPR that different bacteria have adapted to solve this problem of protecting themselves from viruses in new ways. So that means we don't just have a Cas9, there are Cas1 through Cas14 and dozens of other proteins that don't even have the name Cas with a number, they're named something else altogether. And these are all kind of a rich starting point for developing new kinds of genome engineering tools that might solve some of the limitations of the kind of version 1.0 with CRISPR-Cas9. And so really you should think about CRISPR as being this Swiss Army knife of different functionalities, different modalities, each one of which can be applied to different types of applications. Maybe inserting genes, deleting genes, editing single bases in the genome, maybe changing the epigenome, this kind of additional level of regulation. And I thought just to give one example from this past year of an application that's not even gene editing at all, but still relies on CRISPR, it's kind of some really breakthrough work on using other kinds of CRISPR enzymes for RNA and DNA detection. And of course, this has become extremely critical in the last year because of the COVID pandemic. And the first, in fact, CRISPR test or CRISPR device that has been FDA approved is not for gene editing, it's for RNA diagnostics, where a company called Sherlock has repurposed CRISPR enzymes to serve as an even more sensitive detection device for the presence of coronavirus in a biosample than possibly even the PCR tests that I'm sure many of you have donated samples for. So I think with that, I will end. This is, I guess, behind me, you have the view of the downtown Columbia campus, but Dieter and I are both uptown at the medical center, nestled right up below the George Washington Bridge. I didn't really have a chance to talk about my lab's research, but it's been great to come back to Columbia, New York. And yeah, thanks again for inviting me, Francis and Combis, and I will turn it back over to Francis to introduce Dieter.
Okay, that was wonderful. Thank you for breaking down over 30 years of research and making it digestible for those of us that are within the science community, and then also those of us who are just consumers of science. I think that made a lot of sense and was a great setup to what Dr. Egli will be going into next. I do want to pause and say to all those listening at home, there is a question and answer session that will start right after the next presentation. Feel free as you think of questions to just enter them into the box, and then I'll try to get to as many as possible at the end of the discussion. So that's there for you. Without further ado, we're excited to see what's coming next, and I'll pass it on to you, Dr. Egli.
Thank you, Frances, and thank you, Camby's, for the invitation. So it's a privilege to follow Sam, who's also my colleague at the Columbia Medical Center. We have interacted on other occasions, and it's just, I have to say, I'm just struck by how much, you know, research has progressed, and it's really hard to overstate the impact of this discovery, CRISPR-Cas9, and I believe this became abundantly clear in Sam's talk, how transformative this tool really is. I think it's even hard to grasp right now. We're still in the beginning, but the impact that this can have on so many different fronts is just mind-blowing. Actually, something Sam didn't mention, and maybe for those of you who want to go deeper in the field, Sam also wrote a book together with Jennifer Doudna about CRISPR. And as you can see from the title of this, the crack in creation, you know, what really connects well to CRISPR, there's a reason it actually makes a crack or a break in the DNA. So, you know, it really plays on this ability of CRISPR-Cas9 to break DNA. That's kind of its original function. And it is fortuitous that cells repair that DNA, and through that repair, this is when you actually introduce a change. There are some newer tools, which I will touch on later on, that can make variations to this kind of crack or break in the DNA. But what you see here is essentially what Cas9 does. It makes this so-called double strand break. DNA is composed of a double strand, which is drawn here by those two lines. And then Cas9 cuts both of these strands, and the cell then takes this and does this sort of what's called end resection. So it nibbles away some of that DNA and then tries to fill it back in. And you can provide a template. I mean, that can be either another piece of cellular DNA, or it can be a synthetic DNA template that is made in a laboratory. And then the cell will use that to copy it. So it's kind of a copy and paste mechanism. And so you end up with that new strain that has this new DNA sequence, red instead of blue. And of course, this way you can confer, you know, novel sequences, different sequences, and you can repair mutations. So what's attractive about this approach is that it's very versatile. You have the potential to correct essentially any disease-causing mutation. And of course, the first use of this is likely in the adult, in the soma, for people who already have a disease. These attempts are already ongoing, such as for sickle cell anemia, which was just discussed, and for cancer and for other diseases. So this is already happening. But I think what fascinates the public a lot is even whether this will be used to prevent disease, to fix mutations in human reproduction, which, you know, of course, could have, again, transformative effects on us as a species. It has the potential to significantly advance human health. Why? There are millions who are affected by heritable single gene disorders. We all carry genes and genotypes that predispose us to diseases such as diabetes, Alzheimer's, Parkinson's, you name it. And it's diseases that can be polygenic, meaning a number of risk factors plus environmental factors collaborate to bring out a disorder. Of course, it's always meaningful to do something before the problem actually arises, to prevent disease before irreversible damage occurs. Fixing is harder than preventing. It's, you know, the bar to step in and do something is higher. And we often only get into action when the damage is already there and the need really arises to do something. which is obviously not the best way to go about it. So in principle, it would be very good to prevent disease in the first place. And another major advantage to doing that early on, meaning at or around fertilization, that you have the ability to reach and treat all cells of the body. Well, as you can see in that background slide, that's just a single cell. What you see here, this is an egg. And so if you reach that one cell, you will reach all cells emerging from that one cell, which is the entire body. That could have major advantages. In somatic cell therapy, you have to treat billions of cells potentially to achieve the intended therapeutic effect. Furthermore, if it's done in the germline, it will prevent propagation through reproduction. So the solution is lasting. All of these things are, in my opinion, desirable. They could provide a pathway to parenthood without disease for some couples. Of course, there are several ways to accomplish that. You know, like for many things in life, we don't only have one solution to address a problem, but they have each their advantages and disadvantages. So for example, for a couple who has disease-causing mutation, you know, you can do pre-implantation genetic diagnosis, or you can do diagnosis at the fetal stage. In some cases, it may be the way to go, maybe adoption, or a donor gamete may be used. Not all of these options are equally desirable, and it really depends on the case whether they can be applied or not. These down here have not been realized yet. One of them is a genetic change called mitochondrial replacement, which addresses mutations in the mitochondria. This one does not actually involve CRISPR, but this one here is kind of the new emerging one, where to use CRISPR-Cas9 to fix disease-causing mutation in human embryos in the germline. When I'm talking about the germline, that means these are the cells that can give rise to an entire body, but can also give rise to egg and sperm, and therefore go on to the next generation. So there have been quite a bit of thinking about this by regulators or by societies who are interested in, you know, how this one should go about it, whether it would be ethical to do. And so there is a report from the National Academies that came out last fall in September, together with the Royal Society. What they say is that under some circumstances, it may be meaningful to perform germline gene editing, but that the technique is not yet ready. They endorse, however, research on germline gene editing. And I think of a few statements from this report are worth mentioning. What they say is that only common alleles should be introduced into the germline, meaning that, well, if you introduce a common allele, it's something that's already present in the human population. So you're not conferring novel properties to the species. You're not doing something that you don't know the effect of. If an allele is common, then you know it is compatible with human health in various different genetic backgrounds. If you introduce an allele, this is kind of a genetic, you know, variant, a certain genotype. So if you introduce something that is rare, you don't necessarily know what it will cause, right? Because there's not enough knowledge about it. And so this is one thing they're saying, is that only common alleles should be introduced. They say that this technique is not yet ready. It's not appropriate to establish a pregnancy until it has been clearly established that it is possible to efficiently and reliably make precise genomic changes without other undesired changes. What can these other undesired changes be? Well, I'll get a little bit later to that. There is actually quite a few. So these criteria have not been met, but further research is necessary. So they are in supportive of research. And so we are one of the groups actually who are doing such work, and I will show you a little bit of that work. there are certainly enormous ethical challenges about this. These include, but are not only, these are not all concerns, but these include, for one, is equitable access. Even when this technology becomes available, how do we ensure that people of various backgrounds have, access to it? Even today, for reproductive technologies, even now, it's very uneven. Access to these technologies, such as in vitro fertilization, are very uneven worldwide. One major issue is, of course, that parents, who are usually the ones making decisions, the resulting child cannot make any decisions. While parents are autonomous in their reproductive decision, meaning partner choice and various other ways to what they're doing with their lives, it's their decision. The child has no part in these decisions, but so he cannot agree to something. So if a genotype is being changed, then the child has no influence on that. And of course, that could result in exploitation for non-therapeutic modifications, which is often referred to as a slippery slope, or designer babies, which would be designed to essentially have high IQ or designed for certain appearance or athletic skills, or also be designed to adapt to interests of others, including the parents who are having that child. And of course, that's very problematic. So when I talk about these things, I should also mention that we not nearly understand enough about all of these features here to know what to do. So these are not yet actionable, or one could say, well, if you modify this gene that way, the IQ is going to jump 10 points. We don't have that. But as parallel efforts are being conducted at a large scale to better understand the human genome and know what different variants do, I believe that we will ultimately get an idea of what determines these various phenotypes that are, that we have, what determines these features. And so it is possible or conceivable that ultimately there are actionable genotypes that would enable design. So these are important discussions. I personally am strongly opposed, as I'm one doing that type of research. I'd like to say, this is my personal opinion, but I'm very strongly opposed to doing any of the below, and that these powerful tools should only be used to, first of all, understand disease, and second, to advance human health. And I think, my presentation is not going to be very long, so I'd be very happy to, I see the hand was raised, I'd be very happy to answer questions or, in the discussion part. So let me get a little bit about into the research that we've done on that. So we are one of the very few groups, probably worldwide, who have attempted and looked into whether we could repair a disease-causing mutation in human embryos. So what you see here is an egg that is injected with sperm. And then the goal is to repair that mutation and find the embryo that is totally normal. And this was actually first reported from a group in the United States, 2017. It made the front page of the New York Times. Scientists repair a risky mutation in human embryo, milestone in research. So it was worldwide news. You probably have seen it at the time. We wanted to look into that for another mutation that's causing blindness, and that's a gene called EYS. Sounds like eyes. So EYS is a gene. That's the name of the gene. When it's mutated, it can cause blindness. And so one thing that's really fascinating about Cas9 is we have 2 chromosomes in each cell, one from the father, one from the mother. But Cas9 is so specific that it can know that it will only cut the mutant chromosome, not the normal chromosome that would come from, if the father has the mutation, inherits the mutation as shown here, then we can know that Cas9 will just cut that mutant, that the chromosome with the mutation, and not also the one that's normal, right? So we wanted to know whether, as in previous reports, you can restore this normal sequence so that you have an embryo that is entire as 2 wild type, meaning normal copies, WT, that is wild type, so normal, has 2 normal copies of that chromosome and no longer has the mutation. And what we found, very surprisingly, is that this is not what was happening. Even though in certain assays, we would see that, the embryo looked wild type, meaning you could only see this normal copy. So what I should say is that in the mutation, this peak here, which is a G nucleotide, is missing. And when it's present, we know it is wild type. So the lack of that G is the mutation causing the disease. Even though this embryo looked wild type, what it had is it actually missed an entire chromosome arm. So that's shown here. So instead of correcting the mutation here, so this is a chromosome, the Cas9 cuts here, instead of having the mutation corrected, what happened is it simply cleaved off that chromosome arm and the entire chromosomal arm disappeared. So what normally should happen in this plot is that all along the chromosome, we should see those blue dots, which means that both maternal and paternal copies are present. And now we only see those red dots here, which means that the paternal chromosome, which carried the mutation, was actually gone. So that was a new effect that Cas9 could have. It could confer chromosomal changes. It was really fascinating because it was something we didn't expect, that when you cut, that sometimes this chromosome is not repaired. The human embryo doesn't care to repair this break. The chromosome remains broken, and consequently, this piece here of that chromosome is lost altogether. And with that, the mutation, but it wasn't corrected. So there have been a number of studies to date about the use of Cas9 in human embryos. And together, in combination, they do point out a number of challenges, and some of these were mentioned by Sam earlier on. One of them is, for example, off-target activity. So what this graph shows is on the very left, shows repair. How often is it properly repaired? You can see the efficiency is quite low. If you really want to be much higher to be efficient. So the efficiency was more around 5 to 10% across a number of studies. There were mutations or changes, genetic changes that are not necessarily intended. And they were actually more frequent. So they made changes that are slightly unpredictable and that are not genetic variants that are already common in the human population. These are new genetic variants that we wouldn't know the effect of. So these could not, embryos that have any of these, what we call indels, could not be useful for reproduction. Another issue is that these embryos have something that's called mosaicism, meaning different cells have different genotypes. So, clinical application is all about knowing what the consequences are, right? If you pick one cell and see that genotype, you need to know that this is also true for another cell. If you have mosaicism, this cannot be done, right? Because if you pick one cell and the other cell is different, you cannot analyze all cells and also have that embryo for reproduction. So this is also a major issue. Then off-target activity is if Cas9 introduces changes elsewhere in the genome, which is again undesirable. And then we also found it could induce aneuploidies. So all of these outcomes on the right of this first section are undesirable. And you can see some of these bars are quite high. So how on earth could this be done right now? Well, of course not, right? It is not appropriate to do that, but nevertheless, it happened. And I think part of it is maybe not everything was known about the potential detrimental consequences. We often tend to focus on the positive aspects of a story and pick that up, and that registers, but there's really a number of things that need to be addressed first and discussed, and where you also need feedback from peers and colleagues. So this I believe was at the end of 2018. These were these so-called CRISPR babies. What this is that it was done to address the transmission of HIV in reproduction. There are, of course, other ways to prevent the transmission of HIV in reproduction. So this is not essential. So the rationale to do this for therapeutic application wasn't solid. Then what was done, what we know from excerpts from these work, is that the mutation that was introduced is not common in the human population. It was something entirely novel. And so that wouldn't conform with what we have now, which is the NAS guidelines. There was a potential off-target effect. The chromosomal constitution, which we found can sometimes become abnormal, was not evaluated. And then also non-developing embryos were not evaluated. So now we know a lot more. And I would say it's quite unlikely this would be done with the current state of knowledge. We know a lot more about the detrimental consequences than we knew then. But nevertheless, I do think that CRISPR does have a strong future, a potential. What research does in the negative findings of challenges, they narrow the path. So they actually helpful in guiding what is meaningful, but also in guiding what is not meaningful. And so this is a next generation application of CRISPR, which is called base editing. And so what base editing does is was actually developed by David Liu at Harvard University, and here applied, again, a study in China, to the human embryo. So what you see here, this is the mutation. This G, that's the problem causing beta thalassemia. And you see here, when you do sequencing, is that this black peak, that's the mutation. So how to correct that? Well, here is the G, and then it's changed to an A, right? So this is called base editing. You change one letter to another one, and in this case, it doesn't introduce a break into both strands of the DNA. Base editing relies on a break in just one strand. So it's somewhat a little bit of a milder form, you could say, of a break. And this looked very promising. You can see here that the efficiency of these changes is higher than what I showed you before for the double strand break. So it's around 20 to 30%. But even here, there's also some challenges. You can see in addition to this position that was supposed to be changed, you also find some changes that are neighboring and that we don't need, right? That are not necessary. So these are called bystander edits. So that was also found. So while extremely promising, these challenges in combination tell us that for germline therapy, for the therapy of embryos, eggs, sperm, these techniques are not yet ready, very clearly. Nevertheless, people have thought about this quite a lot, whether it's meaningful to have a path. And some countries agree cautiously, others do not. So in the United States, the current situation is even if you had a safe therapy for the germline for a mutation and there's clinical need for it, the FDA could not consider an application. It would simply be ignored. And by ignoring it, in this particular case, it would mean that it's rejected. So it cannot, there is no path to clinical translation in the United States. Other countries do permit the use of genetically, so thus far, there is no single country that has an explicit permit to use genetically modified embryos for reproductive intent. Some have potential exemptions. Most have prohibitive regulation. Many countries have no regulation on research. Some permit explicitly, and a few prohibit. It's about 1-to-1, but most don't have regulation. If those that do, about half prohibit, about the other half permits. So, of course, when there is no path to regulate for clinical translation, this also discourages research, because why would you do research on something that then is not cannot be used, right? It doesn't add up. And so this up here is a very strongly discouraging research to determine if and how germline gene therapy could be used. There is also major challenges at the level of funding such research. So for instance, this is our NIH director, Francis Collins. What he says here is that the NIH will not fund any use of gene editing technologies in human embryos. And the basis for that is really an amendment. And since we are, we would be in Washington without the pandemic, but I think that's why perhaps these regulations may be of interest to some of you. The basis for that is this Dickey-Wicker amendment that says that none of the funds may be used for the creation of a human embryo or embryos for research purposes, or for research in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to a risk of injury or death. And so research of the kind I just showed you relies on either private philanthropy or here in New York, there was a state stem cell program, the New York State Stem Cell Science Program, that tried to fill the gap because of the constraints imposed by the Dickey-Wicker Amendment. Unfortunately, and perhaps a victim of the pandemic, New York State has now just ended their stem cell research funding. But really, what I think gets lost here is that research is really, much of it is serendipity. And there can be benefits that were neither intended nor expected. And I think there's no better example than CRISPR-Cas9 itself. Who would have thought that research on salt marsh bacteria or yogurt will one day cure cancer or sickle cell disease? I certainly wouldn't have. So I think that's something that's something so wonderful about science, that these things can come out of seemingly nowhere. Of course, for those who have worked on these, they may not be that surprised, because they have dived into this highly specific subject, but I was. And I think what research really does, research needs to be really dissociated from whether or not it's becoming clinically useful, because research has a dual purpose. It provides information both to make decisions for use, as well as against the use of certain applications. And we do need to have a factual basis for either of these decisions. And so by the decision not to support on-enable research, which is currently the status quo in the United States for this area, we are also making the decision against having information. And I think that can result to detrimental consequences, because it plays into the hand of those who are not necessarily intending to conform. I think what we found, and I think here again, CRISPR-Cas9 proved to be an extraordinarily important research tool, is that by introducing a break in the human embryo, a break in the DNA, and finding that it often fails to repair it, we now have learned that, and something, an area of research we are pursuing, is that this likely plays an important role in human development and failures in human development. So if you look at human reproduction, it's actually incredibly inefficient. Only about 1 in 4 fertilized eggs make it to, a viable pregnancy. And a lot of the loss is before an embryo implants or early pregnancy loss, even before a miscarriage is felt. So, and some of it is a clinical miscarriage. And this is very, very poorly understood. Again, in part is, of course, regulations that make constraints on research. There's also very little we can do about it. But I think we just found something really important, and we believe that this acts especially early on here, is that the human embryo actually has a lot of spontaneous breaks and in its DNA, and these two are not efficiently repaired. And so as a consequence, the embryo gets lost. But another consequence could be detrimental genetic change that is compatible with viability and leads to detrimental clinical outcomes. And so I think we do need to understand more about that. I think there's great urgency to do research in that area, but there are certain obstacles in place that are not only of the nature of biology and science, they're also regulatory in nature. And so with this, I would like to thank, I had the privilege to work with extraordinary scientists and clinicians who are who are mentioned on this slide. So with this, I would like to pass it back to Francis, and thank you for your attention.
Okay, that also was wonderful. I think it highlighted really well the roles of CRISPR in reproduction, as well as the limitation when it comes to some of the off-target effects that we're seeing, like you described in your Cell paper. I don't want to take up too much of the time from me just talking. It did generate quite a few questions from the audience, so I'll go ahead and start diving into that. One of the questions that we had was specifically for you, Dietrich. They said, so to be clear, you're saying that the 2017 study that made world news did not do what they say they did, question, i.e. correct the mutation in the embryo. And I think this may have been in, I think this question was submitted when you were talking about how sometimes there's loss of chromosomes and that's not actually picked up.
Yeah, that's correct. We believe that chromosome loss played an important role in that. I think, there's always news. Science has to be able to correct itself. And I think this is a good example. It's just you need a number of studies before you move on to the clinic. It is, there needs to be a reproduction of results by different groups and different scientists. I think there needs to be a body of work, but this becomes very challenging when you have restrictive regulation to do research in this area. And I think that enables misunderstandings. It enables incomplete information. So, does this answer the question?
I was just going to add to that. I think what was interesting in this case, and that has been a theme of the CRISPR field in the last couple of years, is how are you looking at the edit? There's kind of early on, there was a very severe observation bias where people are looking at the genome, the effects of gene editing in a very narrow way. And you only see what you're looking at, right? There's like this drunkard's cartoon. If you're looking in one place and there's things happening outside of your field of view, you're not even going to know what's going on. And I think this was a really interesting and important example of if you're looking for editing using a specific approach that will be blinded to some much larger effect going on, like losing a chromosome or making off-target mutations, you're going to miss that event. And I think the field has really awoken or woken up to the fact that you need to take a number of different unbiased approaches to really deeply and fundamentally understand all of the different consequences of a gene editing experiment. And we really have, as Dieter said, a much broader, more comprehensive appreciation of that than we did just a couple of years ago.
So for the original research that's coming out now, are you seeing more so that there's evaluation of chromosomal constitution, which we weren't seeing before, or has that still not quite caught up?
I would say there's definitely a large appreciation. Just the last couple of weeks, there were more studies showing that in some cases, CRISPR-Cas9 can cause large chromosomal loss or rearrangements. But, you know, it's a tougher experiment to look at genome editing in this more unbiased way. And so what's more experimentally or technically accessible is like this focused view. But yeah, I mean, I remember I was working for 1 year in a biotech company when there was a very high profile paper that came out showing that short of a whole chromosome being lost, you could get very large regions of a chromosome being deleted, things spanning thousands or tens of thousands of letters. And at that point, 3 of the CRISPR companies had all gone public and their stocks dropped like 10, 20% in a day, because literally overnight, the field now saw that it's not just small little precise mutations that are being made. You can have major issues left behind by CRISPR-Cas9. And so now, you know, even for somatic therapies, if 1 in a million cells is losing 20,000 letters of DNA, and that chromosome loss falls up against the boundary of an oncogene, something that when disturbed can now cause some proliferative advantage that can cause a cancer, you know, you're going to be in big trouble, even if it's a really, really rare event. So I think, you know, clinically, the companies that are actually advancing these therapies are looking very deeply, because I think for safety concerns, you know, you can't have rare adverse consequences and then put a bunch of gene edited cells in a patient. That's going to spell disaster.
Right. Fair enough. The next question we have, I guess could be for either of you, but I think was from your talk, Dietrich. They ask, how can worldwide research be controlled to keep within any set of ethical guidelines? And I'll piggyback on that and ask, given the limitations in funding currently in the United States, will research in other countries that aren't as tightly regulated also, you know, create a greater push for research eventually here?
It's an interesting question. I honestly don't have the answer to it. How can you control research to keep within any set of ethical guidelines? I mean, I think it needs involvement of, you know, regulation at all levels. I know the WHO is thinking about this. I think it needs, you know, community dynamics among researchers. It needs transparency. It needs, you know, openness. It needs conferences. I don't know how it's possible. How it's possible to make a regulation spanning countries that are intrinsically, you know, bent to set different priorities. I'm sure there are many people on, you know, on this call, on this conference, who would know a lot more than I do. But I think, especially when it comes to clinical, you know, building a consensus is very important. And I think any advance should only occur at the highest level of regulation. I think scientists need freedom to advance their science and, you know, provide information that others can use to make the base decisions on. I think what's very important is that the scientists are not the ones who make the decisions whether or not to go forward with clinical treatment. What happened with the CRISPR babies is that the same people who did the basic research consented, worked to consent the patients, and, you know, decided that it's ready for use and then went ahead. That's never how it should be done. You know, I can do research, I can do excellent research, and obviously I will believe in it, but it is up to others whether or not the work Dietrich Egli did or Sam Sternberg did is ready, right? That's not up to me, shouldn't be. And, you know, that's my opinion, but I think many here on this call will, you know, have something to say about that.
Yeah, I think in 2018, as you were mentioning, we saw where there was kind of an outcry from the scientific community, bioethics community, everybody, really, when we heard about these babies being born and how it seemed quite unethical. And then ultimately that led to some degree of accountability where there were consequences of it. And Sam, I know that I think you had a paper in Science where you guys were kind of proposing a moratorium to have ongoing broader discussions about it. Have you seen a wave now where there's some sort of consensus that we really shouldn't be applying this clinically, or are there still some scientists within the community who are pushing more aggressively for clinical application? That's for either of you.
Yeah, I mean, yeah, I know sometime in 2019 or 2020, there was a Russian scientist that was in the news quite a bit who was speaking publicly about his intentions to do other germline editing experiments clinically for blindness or deafness, I believe. I think, there's not many individuals out there with the megaphone talking about it. Whether or not there are folks, in the fringes thinking about it, I don't know. I don't talk to those people, but I'm sure it exists. I think, I think the interest, I'm sure, is there. It's not going to be embraced publicly by any kind of credible scientific bodies for a long time. But I'll take the more pessimistic take on, can there be international consensus? I would say probably not in like a utopian ideal where we all agree what we're going to do and what we're not going to do, and everyone, obeys the international treaties. I mean, we don't even have successful treaties on far more black and white scientific topics or scientific policies. So, yeah, I think it's tricky. I think it's really tricky. But I agree with what Dieter said. You need to have those public discussions, and you need to have international regulatory bodies like the WHO doing their best to at least bring everyone into the fold and make sure that those brainstorming and ideas are being held transparently.
It sounds like they're largely on the same page. Did you have something that you wanted to add? I didn't want to cut you off there.
Yeah, sure. I think what scientists can do is provide information also what not to do, right? And I think in this case, knowing that you can have large deletions of the way, as I mentioned, this is important. And knowing that you can lose chromosomes, that's important. And I think even people who know that and might not be comply to, that regulations are different in different countries. If they know the facts, they have an opportunity to align their actions with those basic facts. So I think these are actually quite powerful. Having information is, critical, and I see this as our job to provide information.
Sure. We have several other questions. I'll try to get to several of them, but some of the basic science questions. An anonymous attendee asked, how is CRISPR programmed to attach to a specific area of the DNA? And how is the replacement DNA programmed to find where the DNA has been, or the break has been made? You explained some of it. I imagine that it's really going into detail might be outside the scale of tonight's talk, but if you can just quickly address that.
Maybe I'll start. So the information about where to go is this guide RNA, a molecule that's kind of related to DNA and its composition. So that's what tells the Cas9 enzyme where to go. The second part of the question is a very active area of research. How do you increase the frequency of repair by getting that repair template in the right place? And actually one of the newest versions of CRISPR that kind of goes beyond the simple make a double strand cut is something called prime editing. And this was a really ingenious idea where they actually physically couple the repair template to the guide RNA, so that rather than kind of two independent events, first you cut and then you just hope that the repair template gets captured at the broken chromosome. In this new type of prime editing, both molecules are there in the same step, so that one directly leads to the other without any chance for something kind of happening in between. That's, again, a little bit oversimplified, but this has been a really critical area of research. How do you basically help the cell know where to put the repair template when?
Okay. And going along those same lines, there was another question that asked if you could speak about the dynamics of the CRISPR process, like how quickly does the cut/repair take in humans? How fast does it progress through the large number of cells in humans or other organisms?
I guess I'll start with, or Dieter, you should cut me off if I'm just rambling too much, but I love that question. Part of my PhD was focused on exactly these dynamics in vitro, so not in cells, but kind of how can this enzyme sift through 3 billion letters of DNA in any kind of a reasonable time scale? It really boggles the mind, and find one specific 20-letter sequence. And that's kind of the amazing molecular dynamics that proteins have in a cell. They're just jiggling around faster than you can really think about. And somehow they just find the right place. I mean, it's almost magic, I think. But yeah, people have done now very kind of careful studies on these kinetics in cells. The break can be really within tens of minutes of when the protein and the RNA come together. The actual repair can take a little bit longer. I mean, there's a lot of things that have to happen. The CRISPR molecule has to get physically dislodged. You know, building on the last question, the DNA break needs to be recognized. The DNA gets kind of chewed back a little bit. The repair template has to get pulled in. So that can, you know, I don't think we really know on a great level of detail, but I would say it's kind of on the minutes to hours time scale. But then, you know, your question also got into the other side of it, which is now if you're thinking about treating a patient, how fast does it take a delivery vehicle like a virus or a lipid nanoparticle, the kind of thing that's being used for the Moderna and Pfizer vaccines? How does that penetrate the tissue and get to enough cells? And how does that edit propagate if the cells are dividing? So there's kind of a lot of complex things going on there. And I think we don't really have kind of the ideal detailed view yet, but it's another thing that I think from a pharmacology perspective, a lot of these companies are thinking very carefully about. But Dieter, please, you're much more knowledgeable in this area than I am, probably.
Oh, no. I defer to you completely. So, well, I mean, you know, what we see in the, you know, in the early embryo, it takes a few hours, a couple of hours until the cut occurs, and then it takes, you know, quite long, if it happens at all, that the repair happens. I mean, it is indeed, you know, if you think about it, these tools work, these proteins and the RNA, the original biological purpose of it was to cut small genomes.
Yes.
A phage, which is very small. And here you have like, how large is a phage genome, Sam? It's not that large.
Tens of thousands of letters. So we're talking, what is that, 4 orders of magnitude larger genome? I mean, that was a big mystery early on. Like, fine, CRISPR-Cas9 works in a small little bacterial cell on a genome of a couple tens of thousands of letters. Will it work in a human cell? You've got all different kinds of chromosome compaction. You've got nucleosomes. You've got 10,000 times more DNA. I mean, how's that going to work?
I love the enthusiasm. I'm a big fan. It's a very sexy topic, and people are curious about how to move this forward. So I'm going to combine 2 questions here specifically for you, Dieter. One was, what are your thoughts on the most promising and pivotal avenues for accelerating the research in this area, especially given the barriers that you mentioned earlier? And then, do you foresee a way for germline editing to be publicly available for specific cases? And if so, how does such a technology become available given how we regulate biologics/drug products and conduct clinical trial drug development today?
I do see a way. I think that is just informed by what's already here. You know, where do you have solutions that are on solutions already for certain things? Then you're not going to use it for that. But for specific cases, let's say at the moment it's rare, right? Whenever you start with something new, the bar is high. So, you know, you would use it for cases where there is no other option, for instance, dominant mutations, homozygous mutations in both parents in the same gene. So these would be very rare cases. So maybe a couple hundred a year in the United States only. So rare. But these couples, you know, they may want to have a genetically related child and have no other path to it to have a child that is healthy other than that option. So that's where you would start. But these tools keep improving, and the safety profile will change. And you could ultimately imagine that this is being used for risk reduction of common diseases. This is entirely possible. Also, you know, one argument is of course made, well, we'll have somatic gene therapy, we'll just fix things there. And again, you know, you can have, it depends a little bit on how many cells you can access in the adult, how effective it is. But also, if you fix mutations in the adult, you know, for people who would otherwise, you know, make it to adulthood, these patients will then likely want to reproduce, right? So somatic gene therapy does not necessarily reduce the need for germline gene therapy. It may in fact increase the need for it. So these are not mutually exclusive things. And I think, you know, generally in society for important problems, we don't usually have just one solution, right? You have, for energy, you have many sources, and for medical problems, you want to have a tool set of solutions. So I think there is a need. I think there's an appropriate place. I don't think we know exactly what it will be and how this will come about. I think there's much to discuss and much to learn through discussions like this. But I do think there is a way, and I think it will be done. And one thing we will need to see is that we don't put ourselves at a competitive disadvantage without having, you know, a strong reason. I think we need to be open.
Dieter, how many years, if you had to put a guess on it?
That was my question next.
I think at least 10, probably more.
Okay, so I guess only time will tell in that situation. Sam, your wager?
Between 10 and 50, but probably closer to 10.
Okay. Okay. So we'll have to have maybe a 30-year follow-up discussion and see where we are.
No, let's catch up soon.
I will say that, I mean, I love, Dieter, how you started your talk. I mean, the pace of the research is absolutely mind-blowing.
Yeah.
I mean, the CRISPR field, you know, in 2007, like science was pretty damn advanced, but then 5 years later, there was all of a sudden a very mature field of CRISPR biology. And now gene edit, I mean, the world we live in today in terms of what you can do for precision DNA writing is nothing like it was 8 years ago. And so, you know, it's not like a linear incremental progression. That's why maybe 10 years is too conservative. I don't know. I mean, I think probably on the regulatory side, that doesn't have the same exponential growth the way that kind of scientific research can. But, you know, I think a lot about like, what are the different timescales, and it's kind of hard to apply today's thinking on what it's going to look like in 5 years, because the whole paradigm will have changed yet again.
That's fair. And from a policy standpoint, at least being in DC, any kind of embryo research tends to be a little bit controversial. There was a question that came a little bit earlier on. One of the things that was discussed as a potential limitation or barrier was that there may be increased inequality or concerns about equitable access. You mentioned that CRISPR specifically is low cost and there's global distribution, which are some of the pros of it. Does the low cost of CRISPR maybe mitigate some of the concerns about equitable access across the world?
I would say no.
No.
Not my area of expertise, but low cost for research use, you know, clinical development is still a massively expensive operation. So maybe, I don't know. I mean, I would say the equitable access is my biggest personal kind of issue or challenge with these technologies, whether for germline or somatic editing. I just don't know enough about healthcare access and availability to like propose or suggest how that can be mitigated, but I don't think the low barrier to entry for researchers materially affects that. But Dieter, please comment.
I think we're really witnessing a transformation, as you emphasized, Sam. I think it's an absolute privilege to be in this time in both research and ultimately clinical translation. I think, yeah, there are many issues, many things to discuss, but it's still a very exciting time. And I think ensuring that this all comes about, both in the somatic gene therapy, that this comes out about in an equitable way, is very important. I think there's many, that the changes that this can have at so many different levels. You saw how versatile this tool is. At so many different levels, it can really be transformative at so many different areas. I specifically talked about the embryo. You know, the touch is certainly on a very important part of this. But you know, there's many people who will need to think about this, what this means, what the opportunities are, how to help to bring this about in the best possible way. Research has already transformed, but this is only kind of the beginning still.
Right. Speaking of approaches or applications outside of the embryo, more into some of the therapeutic applications that may be in adults or younger children currently, someone asked if either of you are aware of any applications in Tay-Sachs disease.
Dieter?
Tay-Sachs, it can be, you know, currently there's PGD being done for Tay-Sachs that works for many couples who have enough embryos. Sometimes there's just simply not enough embryos around, or the ones that would be, you know, eligible for implantation for the ones that are there, you know, they have the mutation and then they're not used or cannot be used. So for these kinds of cases, gene editing could be useful. In fact, you know, yeah.
Okay. A very practical question came in from an anonymous attendee who asked, so how is this research funded if the federal government doesn't and New York State just cut funding?
Private philanthropy, foundations, donations. That's the one way that still remains.
Okay.
An uphill battle for sure.
It sounds like it. But again, 10 to 50 years, we'll see where we are and where the funding's coming from at that point. Another question, what are each of you most excited about with regard to the potential of your work?
I can start here. So actually, I hope that, well, I guess I see myself thinking more about somatic editing, but you know, where I hope my research and Dieter's will overlap sometime in the future. One thing that we have discovered is a new version of CRISPR that doesn't cut, it actually pastes DNA. And what's really advantageous about that is it actually skips over all of these DNA repair processes that cause the chromosome loss that Dieter just mentioned. And I think that's where the CRISPR field is going. It's kind of moving past making these broken lesions in a chromosome, which is a rather sloppy way of editing the genome, is like breaking it in the process. So we have gone back to bacteria, discovered new natural versions of CRISPR that work in fundamentally different ways than CRISPR-Cas9. And I think from a molecular view, I mean, I didn't talk much about this, but I love thinking about molecules and how they look and how they interact with each other. So we're quite excited about understanding the inner workings of these new CRISPR systems. But from an applications perspective, we think it's going to plug an unmet need right now for how to do gene therapy in a fundamentally more safe and targeted way.
There's a lot of new exciting things coming here. I follow Sam's research as well, so it's always exciting to read his latest papers. For my own research, you know, we are following up on this. What I think is an important discovery is that the embryo doesn't repair DNA very well, and that this could result in, you know, both loss of embryos as well as genetic change that causes disease. And so we want to understand that.
Actually, Dieter,
it will be an important tool to understand this.
I thought that was really interesting. Is there some idea that maybe the embryo, like that's kind of a surveillance approach to ensuring the integrity of fundamental DNA repair pathways, so that if you encounter problems early in development, probably that individual is going to be in trouble later on, and so the embryos evolved a way to kind of surveil for kind of problems with DNA repair early on? Is that what do you think?
It could be some sort of selection. We don't know. It just shows how little we understand about our own species at the very start of life. So there's a lot to learn.
And I think that is the perfect place for us to wrap up for tonight. It's officially 8 o'clock, and we're all here now collectively looking forward to what future work may be coming from both of your labs and the field. I thank you both for joining us tonight. That was a very enlightening presentation and discussion with the two of you. Thank you.
Thanks
Thank you, Francis.
Have a good night.